Our second individual presentation is in the area of computer science. Ivan Paskov is from the Edgemont High School in Massachusetts. His mentor is Dr. Christina Leslie, head of Computational Biology Lab at the Sloan-Kettering Cancer Center in New York City. Ivan.
Good afternoon. My name is Ivan Paskov, and today I want to talk to you about a novel computational tool that is inspired by the way the human brain learns and that has the potential to greatly advance the field of personalized medicine. So currently, cancer is predominantly understood and treated in terms of its organ of origin. However, recently there have been some great advancements with the development of targeted therapies that take into account the genetic variation of a patient. One famous example of these are the genetic tests that search for the HER2 mutation in breast cancer patients. HER2-positive breast cancers tend to be very aggressive, and they used to be very difficult to treat. However, targeted therapies like Herceptin can now cure them. And so to further improve upon such personalized medicine, researchers have begun exploring how to construct computational models that can take into account the genetic makeup of a patient, and from that determine the optimal course of treatment. If we had a computational model that could accurately predict how a patient would respond to a cancer drug given his or her genetic makeup, it would revolutionize the way we treat cancer. Such models would allow therapies to be much more precise, and they would completely eliminate the trial and error process of identifying a drug the patient will respond to, as doctors could now perform a simulation before treatment begins, and from that identify what the patient actually would respond to, and then design a course of treatment that they know would work the first time. And since such models would take into account very fine-grained information about these patients, they could discover similarities between seemingly unrelated cancers to allow us to repurpose existing cancer drugs to fight new cancers. For example, using a lung cancer drug to fight breast cancer. These models therefore effectively multiply out the number of resources we have to fight cancer. And finally, since such models would generally work by identifying the pathways that these drugs target, they could offer novel insights into how these drugs work, their side effects, and potentially how to mitigate them by helping us design new and more effective drugs. And so this is an enormously exciting field, and one in which any progress has the potential to greatly help millions of lives. Current research in this field focuses on predicting drug efficacy in human cell lines. And my research uses the Cancer Cell Line Encyclopedia database, which provides detailed genetic information for 500 cell lines. And for each cell line, the CCLE provides a complete genomic profile with 50,000 measurements on that cell line across 3 genomic categories. We have information on each cell line's gene expression, on their mutation status, and then their copy number alterations. And associated with each cell line's genomic profile is an efficacy value for 24 drugs that tells us how that cell line responded to that drug. And now what we can do is, and efficacy is measured as either an IC50 value or an activity area value. And now what we can do is take the genomic profiles for each cell line and stack them one on top of each other to create a 500 by 50,000 dimensional matrix. And now the goal, and associated with this data matrix, is a vector that contains the efficacy values for each cell line. Now the goal of these computational models is to use the genetic information in this data matrix to predict the efficacy in this vector. And the way we do this is by modeling efficacy as a weighted combination of these cell lines' features. Mathematically, we can represent these weights by the vector WT, the efficacy vector by YT, and the data matrix by X. Efficacy then is approximated by the product of X and WT. And so we are given Y and we are given X, and now we need to find W. And one of the most popular and effective ways to find W is to run an elastic net. And the way the elastic net finds W is by minimizing this equation, which if you notice is made up of 2 main parts. We have the least squares loss and then the elastic net penalty. However, one fundamental problem with the elastic net is that it learns the regression coefficients for each drug separately and is therefore incapable of leveraging the relationships between them. In other words, while the elastic net is learning the regression coefficients for one drug, it might as well not have the information for the other 23 drugs, as it is incapable of using that information to inform its learning. I now propose a novel computational tool based on nuclear norm multitask learning that is explicitly designed to leverage the important relationships between drugs. And I'll start by defining a matrix W, which contains the regression coefficients for each drug as its columns, which makes W a 50,000 by 24 dimensional matrix. And when these regression coefficients are learned independently, as is the case with ElasticNet, W will tend to have a full rank of 24, as the regression coefficients are not constrained to be related to one another. Nuclear norm, however, forces these regression coefficients to be related by constraining the rank of W to be less than 24. Equivalently, it allows us to express W as the product of two matrices, one tall, one wide, each of rank less than 24. And what's nice about this representation is that the columns in each of these matrices have nice biological interpretations. Each column in the tall matrix represents a basis vector that picks out important features that ideally correspond to a pathway that these drugs target. And column T of the wide matrix represents a drug signature for the Tth drug that shows us how to reconstruct the regression coefficients for the Tth drug as a weighted combination of the basis vectors in the tall matrix. In other words, nuclear norm allows us to express each drug as a weighted combination of the pathways that it targets, which is an incredibly useful representation for gaining insight into how these drugs work. And the way we solve the nuclear norm is by minimizing this equation, where the starred term represents the nuclear norm of W, and lambda modulates the relatedness of tasks. However, because nuclear norm learns the regression coefficients for all drugs jointly, this is now a much harder problem to solve computationally than just the elastic net. And so to mitigate this, I efficiently implemented the nuclear norm using the modular optimization framework ADMM. I implemented all the training, testing, and cross-validation functions, as well as the overall optimization engine. And my code is very efficient, and it reduced the typical runtime of weeks to just a few hours, which allowed for fast experimentation. And so the rest of this presentation focuses on evaluating how well nuclear norm can predict efficacy. And the way we do this is by splitting up our data into training and testing splits. Specifically, we first select the random 10% subset of our data and remove it from training as our holdout set. We then use the remaining 90% of the data to learn the regression coefficients, and then using these regression coefficients, we predict efficacy on the holdout set, which can be thought of as representing new data that we wish to make a prediction for. Error is then estimated by comparing the actual drug efficacy to the predicted efficacy. And we use this basic testing procedure in two ways. The first is cross-validation, where we randomly split up our data into 10 groups and then test on each group. The resulting 10 errors are then averaged to provide a stable estimate of the error. The second is bootstrapping, where we select the random 10% subset of our data n times, test them, and then average the resulting n errors to, again, get a stable estimate of the error. And now, using these procedures, I'll present my results. Our first set of experiments determine which measure of efficacy to use. And the way we do this is by first training an elastic net for each drug, and then evaluating its ability to predict on that measure by bootstrapping our samples 200 times. Noise is then simulated by randomly permuting our response variable 1,000 times and then bootstrapping 1,000 times. Our second set of experiments compare the nuclear norm to the elastic net. And here we first tune the regularization parameters using cross-validation, and then we estimate error through another 10 trials of cross-validation. And here are the results for the noise analysis. The y-axis shows a p-value from a one-sided Mann-Whitney test, which compares the mean squared error of the correct responses to the randomized responses. And here a small p-value corresponds to drug data that is significantly different from noise. And as you can see, activity area is a much better measure of efficacy, as for all 24 drugs, its data is significantly different from noise. IC50, on the other hand, at times behaves poorly, and indeed for 70 of these drugs, it is not different from noise. I will therefore present my result on activity area for the remainder of my experiments. This here is the main graph of my research, and it compares the error of the elastic net to the nuclear norm, and standard error bars are provided. And as you can see, the nuclear norm shown in red, it manages to achieve lower error for all 24 drugs. The decrease in error is on average 35% and as high as 61% over the elastic net. And this indicates that nuclear norm's ability to leverage the relationships between drugs is helping it make better predictions. Now I'll provide some biological analysis of nuclear norm's predictions. If you recall, we could express W as the product of two matrices, one of which provided us with drug signatures. And we perform clustering analysis on these drug signatures and expect that drugs that have similar mechanisms of action should have similar drug signatures. And these colored boxes here indicate drugs that target the same pathway. And amazingly, nuclear norm manages to identify all 4 major groups of drugs in the CCLE database, which is an excellent indicator that it is learning to predict efficacy based on the pathways that these drugs target, which is an incredibly exciting finding. We also perform this clustering analysis on the elastic net, but as you can see, the results aren't very good. The clusters tend to be very flat, which indicates it isn't picking up rich structure in the data. And even worse, related drugs tend to be far apart in this graph, all of which brings into question the validity of elastic net's predictions. And so in conclusion, I propose a novel computational tool based on nuclear norm multitask learning to predict drug efficacy. I show that activity area is a more robust measure of efficacy than IC50, and predicting on activity area, nuclear norm manages to outperform the elastic net by 35%. And this increase is further substantiated by the fact that its predictions are not only accurate, but biologically meaningful, as shown by our clustering analysis. And this shows us that its ability to leverage the relationships between the drugs is helping it make better predictions, and it is therefore a fundamentally different approach from the elastic net. Interesting future work would be to further explore the biological relevance of nuclear norms predictions through gene ontology enrichment analysis, and preliminary results from that seem very promising. And so in conclusion, nuclear norm multitask learning represents a significant step forward for personalized cancer treatments, and I believe it should be a standard tool in cancer research. And to help facilitate that, I plan to release my code so anyone can begin using it. Finally, I'd like to thank my mentors, Dr. Christina Leslie and Dr. Alvaro Gonzalez at Memorial Sloan-Kettering Cancer Center, the Science Scholars Program, and all the teachers at Edgemont High School that helped me prepare, my family for their support, and of course the Siemens Foundation, the College Board, and George Washington University for providing me with this wonderful opportunity to present my research. And thank you to the judges for your time. Here is a selected bibliography, and that concludes my presentation. Thank you.